Brian, should I just get going?
Yeah, go ahead. Floor is yours. Thank you for joining us.
Sure, yes, it's my pleasure. So I'm going to talk about metabolic
Good.
phenotypes.
Okay, well, take care of yourself as well. All right.
I'm trying to reach Celeste. Hey, Celeste.
Oh, you know
Celeste,
what?
are you there?
Hold on one second.
Yeah, don't worry about it at all. Okay.
Be able
All
to,
right. See you.
I wonder if I
Bye.
have the power to mute. Let's see.
You might. Celeste, can you hear me?
I can.
Oh, sorry. You were unmuted. We were hearing your conversation. I was just going to ask you to mute. It's okay.
Sorry, I'm in the wrong direction. Apologies. I
You're
hope
good.
it's too sorted or anything.
No, not what we heard. Interesting. But, I'm not
I
too
heard
sure.
you call Ralph being a very difficult person.
Well, Ralph and I share enthusiasm for sports, so we're on the same page there.
We do, and that can be difficult. But yeah, it's good to see everybody. So I'm going to talk about metabolic phenotypes and cancer progression in humans today. And I know the primary interest is in kidney cancer. I'm going to spend a little bit of time, I hope not offending you, by talking about lung cancer, just to provide some comparisons and I think interesting contrasts to what I'll talk about in the second half in kidney cancer. So my lab is primarily interested in how mutations cause disease through effects on metabolism. And we study two different types of disease that follow that paradigm: inborn errors of metabolism, which are germline mutations in metabolic genes that ultimately impair growth and development, and cancer, where mutations, mostly somatically acquired, promote pathological cell growth. And that occurs in part by reprogramming metabolism. And so about half of the lab studies each of these type of diseases. And I think they integrate in interesting ways. I'm only going to talk about cancer today, but almost everything that we do in cancer on some level is informed by the fixed metabolic phenotypes that we observe in patients with these interesting germline mutations. So cancer is a genetic disease, and it is associated with differences in metabolism at the cellular level. I think one very obvious example of that is the fact that FDG PET scans are useful in clinical oncology. This is a lung cancer patient, and you can see that the tumor tissue here, of course, is taking up and retaining this radioactive labeled analog of glucose. One of many examples that is clinically relevant, where the metabolic changes in tumor tissue can be exploited. Okay, so why do tumors behave that way? That's complicated, as you all know. This is at least in part because of cell intrinsic differences in metabolic activity dictated by gain of function mutations in oncogenes, several of which you see there. And probably the most famous and the simplest to observe metabolic change downstream of oncogenic signaling is what we call the Warburg effect, where there's an increased propensity of malignant tissue to take up glucose, convert it into pyruvates, and then into lactate, and then secrete it. And the dogma for a long time was that came at the expense of oxidizing pyruvates in the mitochondria in pathways like the TCA cycle, which is quite interesting because in most tissues, the TCA cycle is the primary source of energy, of precursor molecules for macromolecular synthesis, and is important in redox balance too. So I've been in the cancer metabolism field for about 15 years now. And I thought there have been a couple of really interesting aspects of this classical paradigm. One is that because there's this convergent property of enhanced glycolysis downstream of many different oncogenes, that you could get the perception maybe a couple of decades ago that most tumors would look essentially the same metabolically, because you could draw a pathway like this that would occur in downstream of many different kinds of cancer driven by several different oncogenes, and that would more or less be correct. Okay, so sort of a monolithic picture of cancer metabolism. And the other is the curious question of whether mitochondrial metabolism was really detrimental to cell growth. That was clearly the dominant view not that long ago. And that seemed interesting because of all of these different aspects of mitochondrial metabolism that are important in non-malignant tissues. So my lab has been basically studying those two questions ever since then, and those will be themes of what I talk about today. So we use two different complementary approaches to study metabolism called metabolomics and isotope tracing. They both use mass spectrometry, but actually they're quite different conceptually. Metabolomics is sort of like taking a picture of a parking lot and counting up all the different types of cars in the parking lot and comparing what's there in two different states, say on Monday morning versus Saturday afternoon. Okay. In metabolomics experiment, you would extract metabolites from a tumor, from the adjacent tissue, or from two different types of cancer, and catalog all the metabolites and try to determine what's different. Incredibly data rich, very informative type of approach. Doesn't give you all the information about metabolism though, because the metabolites will flux along metabolic pathways, just like the cars will drive out on the roads. Okay. And so this is just a still shot of a highway and an off ramp to demonstrate why having some information about pathway activity is also important. You can see all the cars here on the off ramp. And so if you're just basing your assessment of metabolic activity based on the abundance of metabolites in a pathway, you would conclude that this is the active pathway here, when in reality, the flux has been stopped by a traffic light, and actually the active pathway is down here. So we need to have another way to try to assess relative activity of pathways. And the way we do that is through isotope tracing, where we take a nutrient of interest like glucose, tag it with an isotope that can be followed. All the data I'm going to show you for the rest of the talk is using stable isotopes like carbon-13. That is a heavy form of carbon that does not undergo radioactive decay. And that's useful because it means you could administer relatively large amounts of it to either experimental models of cancer or, as you'll see, even in patients. What happens then is that the tagged nutrient will go into the tissue, and that tag, shown here as the red asterisk, will get distributed to downstream metabolites as a consequence of the metabolic activity that's happening in the tissue. And then you can extract the metabolites and use mass spectrometry or other analytical methods to assess where the carbon-13 went, and then use computational approaches to try to recreate the metabolic pathways that led to the labeling phenotype. Okay, so let me address those two questions. The first is whether cancer cells are really monolithic in their choice of metabolic pathway. And we've studied this many different ways over the years, but I think the maybe the clearest reason why we think the answer is no came out of this paper. So a student, patient Chen, in my lab, together with Ling Tsai, bioinformaticist, took over 80 lung cancer cell lines, all from non-small cell lung cancer tumors, in collaboration with John Minna, and cultured them under identical conditions in the lab, and then probed the metabolism of those cell lines with either carbon-13 labeled glucose or carbon-13 labeled glutamine, where the only thing that was different in the culture is where the carbon-13 was, and then extracted the metabolites, read out where the carbon-13 went. And because the Minna lab had carefully annotated so many different biological molecular properties of these cell lines over the years, we thought we would be able to define metabolic heterogeneity across the panel and then search for associations with things like oncogenic drivers and gene expression signatures. And so I just show you an example of the kind of data. This is all published in this Molecular Cell paper from a couple of years ago. And if you like looking at metabolic data, all of the data from all these 80 plus cell lines is in a very user-friendly web app that you can get through that paper. And so what I'm showing you here is the correlation of two different metabolic features in this metabolite, citrate or citric acid, which is the entry metabolite in the TCA cycle. Okay, very simple metabolic property. Almost all cells that we've ever analyzed have the ability to make citric acid. It's a key precursor for lipids. And what you're seeing here are basically two different pathways, this red pathway and this blue pathway that lead to citrate through very, very different mechanisms, starting with different nutrients. And the position of the dot, each one of which is an individual cell line, basically tells you whether they're using more of the blue pathway or more of the red pathway. And the key point from all of this is just that the cell lines are all over the place in terms of their preference for being blue or red in terms of the ability to make citric acid. So hopefully you can appreciate that. And then that turned out to be related to an intrinsic property of the cell lines, because cell lines with EGFR mutations tend to be over here towards higher values for the blue pathway. And that's because gain of function mutations in EGFR stimulate this pathway by activating the enzymes that allow citrate to be formed in this fashion. And those enzymes specifically are IDH1 and IDH2. And we studied this with Jing Chen's lab at Emory in a recent paper. Okay, so sometimes cell intrinsic properties really dictate metabolic preferences, but cell lines are quite heterogeneous in their preference for using various pathways, even to do things simple like making lipogenic precursors. Okay, so we still study a lot of cancer metabolism using cell lines in the lab, but I'm sure everybody realizes that we can't model all properties of tumor metabolism in culture because actual tumors are complex. They contain mixtures of malignant and non-malignant cells. And furthermore, all of these properties shown on the right have the capacity to alter metabolism. And not all of them are cell intrinsic, right? A lot of them have to do with the cancer microenvironment. And other factors that we can't recapitulate very easily in the culture dish. And so what that says is that we need ways to probe tumor metabolism in an intact tumor in vivo, and preferably in patients. So I'm going to spend most of the rest of the time talking about our clinical programs, trying to probe cancer metabolism using these non-radioactive, stable isotope metabolic tracers in different forms of cancer. And I'll start with lung cancer and then move into kidney cancer after that. So this just shows the principle of these studies. We identify patients, for example, non-small cell lung cancer patients, that are going to have their tumor either surgically biopsied or resected. And then on the day of the surgery, we place an IV and infuse a bag of carbon-13 labeled glucose or some other isotopically labeled nutrient. It circulates through the body for 2 to 3 hours. And then that allows the label to enter the tumor, being carried by the tag nutrient. And like I explained before, the tag, that is the carbon-13, then gets distributed to downstream metabolites as a consequence of the metabolism happening in the system. So the tumor is removed by the surgeon. In these studies, the surgeon is Kemp Kernstein, a fantastic collaborator for the last decade plus. Usually the tumor is removed along with some adjacent non-malignant tissue. We isolate the metabolites and then assess the carbon-13 labeling, just like I showed you before in cell lines. Compare the labeling between the malignant and non-malignant tissue. And then over time, we try to follow the outcome so that we can find aspects of the metabolic phenotype informed by carbon-13 labeling that correlate with subsequent aggressive behavior of the tumor. So I'm going to just show some data from patients that were infused with glucose in which all 6 of the carbons were replaced with carbon-13. Just again, a heavy form of carbon, whereas the untagged, naturally abundant form of carbon would be carbon-12, and we're just basically adding 1 mass unit. That's shown by the black dots. And by tagging all of those different carbons, it allows you to follow the fate of those carbons through the glycolytic pathway here, or the so-called Warburg effect, or transfer of the carbon-13 into the TCA cycle through the pyruvate dehydrogenase reaction. Okay. This is the aggregated data from about 70 patients that have been infused with carbon-13 labeled glucose using this setup. And this is complicated, but I'm going to walk you through what this figure shows. Every dot is a fragment of tissue from one of the patients. The pink dots are from the tumor and the blue dots are from the lung. And these correlation graphs here are correlating the amount of carbon-13 in one metabolite related to the TCA cycle, for example, citrate, against another metabolite in the same pathway, for example, malate. Why do we do that? The level of carbon-13 enrichment you get in these studies is relatively low. And I've always thought that this could be subject to artifacts. And so it's important to determine whether the labeling of different metabolites along the same pathway are related to each other. That is, if they're correlated with each other. Because if they are, it greatly increases our confidence that we're reading out bona fide activity of the pathway. And so for all of these different correlations, you can appreciate the correlation is quite high. And that means that despite the low fractional enrichment, the low fraction of each metabolite that contains carbon-13, we're reporting activity of the TCA cycle. And in fact, this cycle turns over. And so you can follow it through 2 and 3 and 4 rounds. And every time you do that, the labeling features correlate well. So that's good. Reassuring from an analytical point of view. Second, you can see that there's quite a bit of variability, right? I mean, both in the adjacent lung tissue and in the tumors, they're quite variable. Okay, so that's useful to know too. Obviously, the clinical outcomes of these patients will also be variable. So we like to see some variability because it gives us a chance to see what correlates with the outcome. And then third, and I think most obviously, for all of these metabolites, the fractional enrichment is rightward shifted in the tumor relative to the lung. You can see that in the distributions here. And so what that tells us is that carbon from glucose is making more of a contribution to the TCA cycle in the tumor tissue than it is in the lung tissue. And so that is not really consistent with a suppression of the extent to which carbon from glucose enters into the TCA cycle. Okay, so that pathway is active, and it's heterogeneously labeling these different tumor tissues, but it's labeling the tumor more than it's labeling the adjacent lung.
Hey, Ralph, is it okay
Yes.
to ask questions?
Please do.
So are you surprised by this level of fractional enrichment overall, or is this what I appreciate that the tumor is more active, which is what we might have imagined, but would you have anticipated this kind of fractional enrichment?
This is sort of about what I would have anticipated. If you look at, most of these are peaking out around 15 or 20%.
Mm-hmm.
Okay. And the way we infuse the glucose, the fractional enrichment of the circulating glucose is usually around 40.
Mm-hmm.
So that means that the maximum amount of enrichment you should get in the Krebs cycle intermediate should be 40. We're getting about half of that. There's many different nutrients that can feed into the acetyl-CoA pool.
Sure.
So it's not surprising that you get some dilution of enrichment in the TCA cycle relative to the precursor. So this is sort of about what I would expect.
Okay. I guess if thinking about that way, it makes sense. I was just looking at the y-axis and seeing things like 0.04 and which seemed a little bit lower than I might have imagined. But if the best you could do is 40%, then if you're getting up to almost 20%, that's actually pretty, I would say, pretty good labeling then.
Yeah, it's decent. And, this is really reporting the M+2 fraction. And so as the carbon continues to cycle around, you end up getting different labeled forms in addition to M+2.
Right.
And that is probably also giving the appearance of a low enrichment. But having said all that, I don't think, I'm drawing the pathway really simply, right, with glucose feeding into the acetyl-CoA pool. But as you know, there's a lot of different arrows on here of unlabeled nutrients feeding into this metabolic pool that I'm not showing.
Right.
And, there's probably a couple of very significant sources of acetyl-CoA that we haven't probed in this pathway yet. And one would be fatty acid oxidation, which could potentially be a big source of acetyl-CoA that we haven't really captured yet.
Also acetate itself, right?
And acetate itself, very good point. Yep.
Okay. Thanks.
Yeah, thank you. Okay, so
So
I
could I
mentioned,
ask you-
Sorry, can I ask you another follow-up?
yeah, go ahead.
So uh, this is great when you see the correlation between the pathway intermediates and it gives you a near linear picture. But what are your thoughts on experiments where you don't see this happening, where 5 out of the 8 metabolites go in one direction versus 2 or 3 of the other of the metabolites now start behaving in a anomalous way. How and I know obviously one way is to start rationalizing based on whatever biochemical knowledge we have, what else could be feeding into the pathway, but where does the hypothesis fall apart?
Oh, well, I think if we saw no correlation with these metabolites anywhere in the pathway, I would think there was an analytical problem with the way the carbon-13 was enrichment was calculated. And we do see that sometimes. I mean, often what happens is that's a signal that there's some interfering metabolite on the mass spec that you have to go in and figure out. And then once that gets cleaned up, usually the correlations get better. If you see a pattern where you have correlation across part of the pathway and then lack of correlation, or even more interestingly, a negative correlation with other metabolites, that suggests that there's something very different about the pathway. And once you've ruled out technical artifacts, it could be a suggestion that part of the pathway is either being fed by a different nutrient, specifically at the point where the correlation falls apart, or potentially even being reversed. And, over the years, we've identified interesting aspects of metabolism simply by looking at where the correlation breaks down, particularly where, like, if you have nice enrichment for part of the pathway and then a significant drop off, that suggests there could be an unlabeled nutrient coming in at that step. And then by going back to the metabolic pathways, you can sometimes figure out how that happens. Okay, so I mentioned that it's nice to have some heterogeneity across the population, because that allows you to try to correlate with outcomes. And so, in this study, through metabolomics and isotope tracing, we had hundreds of metabolic features from each tumor. And in comparing the correlation of all of those different metabolic features with outcomes, the one that really stood out is actually the labeling of the TCA cycle intermediates, which is why I've been perseverating on this, where the tumors that have the highest enrichment in Krebs cycle intermediates, those patients do significantly worse in terms of recurrence-free survival, or as shown here, overall survival, mostly because they die of metastasis. And so what this Kaplan-Meier curve is showing, this is very simple. We took all the patients, okay, and just split them into ones that have higher than the mean level of enrichment in the TCA cycle versus lower. And just simply by splitting into those two different groups, you can see significantly worse overall survival in the ones whose tumors have high enrichment in the Krebs cycle intermediates. So I'm not making any statement about what that means yet. I'm just saying that however we slice this, in terms of assessing all the different metabolites, considering the Krebs cycle enrichment as a continuous variable versus just splitting into two halves, it always basically tells us that it's worse to have a tumor that likes to use glucose as a carbon source for the TCA cycle in non-small cell lung cancer in patients. But I think that's interesting because, remember, the infusion is happening way back here. And then sometimes it's months or years before we see clinical evidence of disease progression or metastasis in the patient. And yet something about that activity seems to be predictive. And so that's certainly worth studying further.
And sorry if you mentioned this already, but is this just bulk tumor that's being assessed, or is it specifically the tumor cells that are being then isolated?
No, these are bulk tumors.
Okay. And so is there, I guess I was going to ask, is there a sense that this is definitely being driven by something tumor intrinsic, or conceivably could you have a active myeloid population that's infiltrated, or some active fibroblast population, something like that, that just, might be tumor extrinsic?
Hold that thought.
Okay.
I will say that, we are very interested in that question. I think the technical ability that we've been able to follow the kinds of metabolic features that we get with carbon-13 tracing, any kind of disaggregation in the tumor has made it really difficult to get at cell-specific properties. I mean, I'm sure everybody on the call knows the beautiful paper from the Rathmels about using radioactive isotopes in mouse models of kidney cancer that really convincingly shows that there are different preferences of immune cells versus cancer cells for different nutrients. In terms of the carbon-13 studies, we haven't quite gotten there yet. I will say that, we look at every fragment of tumor for stromal content and for immune cell content, and no story has emerged that suggests that these properties are related to the metabolic, are more related to the metabolic activity of the non-malignant components. And through single cell analysis, if you look at the gene sets that correlate with this Krebs cycle intermediate labeling, you get TCA cycle genes and OXPHOS genes, which makes sense, but actually through single cell analysis, it looks like those gene sets are most represented in the malignant cells opposed to the non-malignant cells. So my guess is that this is a malignant specific metabolic phenotype, but what we would love to be able to do is to read that out directly in the kind of experiment that you mentioned. Okay, I do want to talk about kidney cancer too, so I better pick up the pace a little bit. Just to make the point of why we care about activities that seem to mostly correlate with what will happen later to the tumor as opposed to its activity in the treatment naive locally invasive state, is just this principle in the field that I think has become more prominent over the last few years from many different labs, that the metabolic preferences and vulnerabilities of cancer cells change as the cancer progresses along either of these two pathways, either the acquisition of therapy resistance or metastasis. And I'm not going to go into too much of the background data. I'll just say that there's lots of evidence from Joan Brugge, from Felice Papianakopoulos, from Sean Morrison and others that a significant bottleneck to metastasis is the ability to deal with reactive oxygen species because of the oxidative stress that seems to accompany loss of attachment from the primary tumor. And so there are multiple papers from various labs, including Sean's, that show that this is potentially a targetable vulnerability. At least as a way in experimental models to call potentially malignant cells out of the circulation. Okay, so of course, I think we're all interested in properties that are important in tumors along either of these two branches, because these are the states where the cancer tends to kill the patient. Okay, just one brief comment then about this apparent connection between TCA cycle labeling and disease progression in non-small cell lung cancer. We would like to think that that's related to something that to the electron transport chain, because normally the way the TCA cycle functions is that it turns in concert with an active electron transport chain or oxidative phosphorylation. That's because as the cycle turns, you get reduced electron carriers like NADH, which then get recycled by the electron transport chain. So normally these act in concert. That'd be valuable to know, because there are at least some tool compounds that have been developed for clinical trials, like this complex 1 inhibitor, this IX compound that was developed at MD Anderson. Okay, so if you thought that the Krebs cycle turning was based on having electron transport chain activity in the cancer cell, you could potentially try to target it. It's important to realize, though, that there are other ways to reduce these electron carriers without using the electron transport chain. So it remained a question, at least in my mind, about whether we actually had oxidative phosphorylation happening in the cancer cells. And so Penny, a postdoc in the lab, decided to take this on by basically infusing mice with xenografts, okay, of various different cancer types, infusing them with carbon-13 glucose, either after being treated with DMSO or with this electron transport chain component. If the Krebs cycle intermediate labeling requires electron transport chain activity, then the labeling should go down when treated with IX. And you can see that he gets this really nice reduction right here. Now, if that involves oxidative phosphorylation in the cancer cell specifically, he should be able to overcome that by expressing a yeast protein called NDI1, which has been used extensively by Nav Chandel's lab and other people to try to take apart various parts of the electron transport chain activity. And that just recycles NADH back to NAD plus and takes the reducing equivalents and puts them in CoQ. So basically it allows cells to maintain something like electron transport chain activity, even when complex 1 is blocked. Okay, and so by putting NDI1 only in the cancer cells, recreating the xenografts and doing this experiment again, now he gets no loss of labeling with the complex 1 inhibitor. And that indicates that at least in these models, okay, it's electron transport chain activity in the cancer cell itself. Okay, that's accounting for labeling in the TCA cycle. Okay, I'm going to run through the next couple of slides very quickly, just to say that we think oxidative phosphorylation actually is important for metastasis of non-small cell lung cancer cells. We've used Sean Morrison's approach to track metastasis in PDX models. Brandon, together with Aslan from Sean's lab, tried to establish non-small cell lung cancer PDXs from 50 patients. He got 11 of them to grow. 10 out of the 11 came from patients that had high carbon-13 labeling in their tumors. Okay, those tumors were luciferase tagged and then reimplanted so that they could assess metastasis either by bioluminescence or by human HLA expression. And they do metastasize. Okay, so this is showing two different models here. The fraction tells you the fraction of tumors, of mice that had these tumors in the flank that had HLA positive cells, some in a different organ. Okay, so these are human derived cells that are viable in the liver, the lung, the brain, et cetera. You can see most of these mice have these viable cells. And the only one of the 11 that doesn't is this one here, 57M. This tumor does not metastasize as far as we can tell. That's a special case because it was the only tumor that came from a less labeled primary tumor in the patient. And it came from the same tumor that gave rise to this 57C. That patient had a large tumor. It was metabolically heterogeneous. Brandon established PDXs from the well-labeled part of the tumor and the poorly labeled part of the tumor. They both established PDXs in the flank, but only one was able to metastasize. Okay, so just to pause there for 1 second and say, it's been very difficult to get non-small cell lung cancer PDXs to spontaneously metastasize in the mice. So I already think that this is potentially useful. And of course, it gives us a way to check whether this is related to oxidative phosphorylation. I'll just say, it's one thing to have a few human HLA expressing viable cells in these end organs. It's another thing to have a symptomatic metastasis. And we are occasionally getting symptomatic mets too in the mice. This is a patient whose tumor metastasized to the brain. This became clinically apparent a few months after we did the infusion in the patient. And mice that are carrying that tumor in the flank occasionally get neurologic phenotypes like this mouse that's incessantly circling to the right here. Okay, that can be a sign of a brain met, and in that mouse, it is a sign of a brain met. Okay, so here's a histologically and clinically apparent brain metastasis in this mouse. Okay, so we see that in two different models that were derived from tumors that gave rise to brain mets in the patients too. So does any of this require oxidative phosphorylation? So Brandon implanted tumors in the flank, treated the mice with DMSO or the complex 1 inhibitor, and basically found that there was not a statistically significant effect on growth of the tumor in the flank, but in the IAX treated mice, there is a significant reduction in the fraction of viable cells in the circulation, and more importantly, a reduction in metastasis, shown here in the brain based on the fraction of human HLA positive cells in this model, and here in the lung, this is now bioluminescence flux in a BLI experiment, and this is about a 10-fold reduction in the overall average metastatic load. We're not sure yet if this is related to oxidative stress, but those experiments are ongoing. I do think this says that there's something about oxidative phosphorylation that seems in these models to be more important for metastatic spread than growth of the tumor at the primary site. Okay, so let me summarize and just say that the carbon-13 infusion protocols are versatile and they can be informative. And so we've studied these in a number of different types of cancer now, some of which are published. And this idea that carbon from glucose in the circulation ends up getting into the TCA cycle in the tumor, it seems to be the rule rather than the exception, because we've observed that in several different types of cancer, and it's also been observed in some nice studies in triple negative breast cancer reported by the Rabinowitz lab last year. There's a component of the labeling that is a little more complicated than what I've shown here, because some of the label that ends up in the tumor's TCA cycle comes in as lactate produced from glucose elsewhere in the body. The tumors, some of the tumors have the capacity to take up lactate and use that to feed the pyruvate pool that ultimately gets into the TCA cycle. And we think that's relevant to metastasis too, but I'm not going to go into all the details of that right now. This is an example of something that Abhishek mentioned before, which is that sometimes you see labeling patterns that don't quite make sense. This capacity to take up lactate from the circulation, we were clued into that by labeling patterns that didn't quite make sense. So it really, it's worth the time to really think carefully about what the labeling is telling you. All right, so lots of tumors that have this glucose oxidation phenotype. There is, of course, a tumor that you would hope doesn't look like that, and that's kidney cancer, specifically clear cell renal cell carcinoma. I think because of the complexity of analyzing the data and how difficult these intraoperative experiments are, it would be reassuring if we got the expected result in clear cell renal cell carcinoma because of what we know from preclinical models and also clinical studies that Celeste Simon and Ari Hakimi and Ed Resnick, et cetera, have done in the past, mostly through metabolomics, that really do suggest there's suppression of the TCA cycle. Okay, so I'm going to spend the last part of the talk telling you about what we've been doing in clear cell. So, why do we think that the TCA cycle should be shut down in these tumors? Well, it's basically because of the physiologic role of VHL in suppressing the levels of the alpha subunits of HIF transcription factors. Okay, everybody knows that clear cell renal cell carcinomas usually lose VHL function, and that means the HIF alpha subunits are chronically stabilized and can put in place gene expression programs that, among other things, reprogram metabolism fairly profoundly. And one of the well-validated targets of HIF1 is this kinase PDK1, which phosphorylates and inhibits part of the pyruvate dehydrogenase complex. That's the very enzyme that allows carbon from pyruvate to get into the TCA cycle via acetyl-CoA. Okay, so if all of the beautiful work from the preclinical models is relevant to human tumors, we should see a suppression of this pathway in these tumors. And this is all data that comes from one intrepid and very talented PhD student in my lab, Divya Beswada, in collaboration with Vitaly Margulis in urology. And so together, Divya and Vitaly have probably infused about 70 patients with different types of kidney cancer at this point. And I'm showing you some of the data here. This metric is the fraction of the citrate pool that contains carbon-13 relative to the amount of labeling in pyruvate in the same tissue. Okay, so basically this is a readout of the relative ability of pyruvate dehydrogenase to transmit carbon-13 from here down to here. And you can see that these are variable, but there's a nice suppression, 50% or more in treatment naive primary clear cell renal cell carcinomas in the kidney of the patient. This is the exact opposite of what we saw in non-small cell lung cancer, when this metric was about twice as high in the tumor as it is in the adjacent lung. Here it's about half as high as it is in the adjacent kidney. Okay, and it's quite variable if you look at all these other different types of kidney cancer, but at least there's this nice suppression that we would have expected to see in clear cell. Okay,
Rob,
and
Another quick question, if we could
Please.
go back one. What surprises me is that CCRCC isn't exactly the champ, right? Like the papillary version looks even more suppressed, and that's not known to have VHL mutations. Maybe you guys know why that is, or do you not have enough tumors to really tease that out?
I don't think we have enough tumors to figure that out right now. I don't know why that is. I agree with you. That's a little bit surprising.
FH or SDH. That's what struck me also, Celeste. So maybe FH or SDH mutation.
Yeah.
those.
So thinking about it,
Type 2
because we're looking at
papillaries will have
citrate, right? We're
fumarate
looking at-
hydratase and
goes all the
succinate
way around.
dehydrogenase
Right.
mutations. So that's
Uh,
probably what's resembling the phenotype over here.
Well, that's a good, very good thought. What I'm not showing you here, because it's even harder to explain, is that we do have a number of FH deficient tumors that we classify differently as papillary type 2 or HLRCC. And those look more like the chromophobes, where there actually is not a significant reduction. Okay, I can't explain this yet either, but those FH deficient tumors really look in vivo as if they're able to cycle the carbon past the FH block. And so we're speculating that there is a detour that involves reversal of enzymes that use fumarate as a substrate in other pathways. And the first part of that mechanism has been described really well by Christian Frezza and Al Gottlieb and Patty Pollard. The second step that would allow the carbon to get released and come back into the TCA cycle has not been described yet. And so I'm not sure if that's what explains that phenomenon in those tumors, but we're working on that. In terms of the papillaries, I mean, really, Celeste, I should put the N's on here. The number of patients that we study with CCRCC is probably 10 times higher than papillary.
Yeah,
And so it's possible that what we're reading out here is low labeling in the papillary tumors is a consequence of the fact we just don't have enough tumors yet.
I see. Okay, thank
Could
you.
be.
Even the oncocytomas though, right? So Vamsi Mutha's stories about the mitochondrial subunits of several of the ETC components being mutated there. These accumulate mitochondria like probably 10 times more than even normal kidneys do. And functionally, I don't think we understand what's happening in those. So even the oncocytomas are kind of interesting for me because I might have predicted that they would actually be higher than the adjacent kidneys in terms of their TCA cycling. But the fact that you're seeing, again, I don't know what the numbers are for that sample, but if that decrease is consistent, it would be interesting to ask what is the genetic phenotype underlying that.
Yes, I agree. And so the oncos are really interesting because they have mitochondrial defects, but then they try to compensate for that by massively increasing the amount of mitochondrial DNA. And so, the question is where they're going to end up. And here they end up as being low, but not the lowest. So I agree, it's kind of interesting. I should point out that there is a difference between isotope tracing and true flux analysis. This is a single time point, okay, when the tumor comes out. And so what we're reading out here is some metric of the success with which the tissue can get carbon from glucose into this pool. And in this case, this part of the pathway right here between pyruvate and citrate doesn't really tell you about the rate. And so you can imagine a situation where, the PDH activity is not actually any difference, but the fraction of the citrate pool that arises from some other non-glucose dependent pathway like fatty acid oxidation, for whatever reason, is massively increased in the tumor, that would read out as a reduced labeling ratio. And so we always have to be open to the idea that we're not actually reading out a dysfunction of mitochondrial activity, but it's a consequence of the way the experiment is done. So, you usually need more information. And so let me tell you a couple of pieces of additional information that really make us pretty confident that this is caused by a pretty complex abnormality of mitochondrial activity, at least in clear cell. Okay. Well, one other question we ask is that, because, you know, the surgeries are complicated. We want to make sure that there's not simply an artifact associated with the surgical intervention that's giving a difference in apparent metabolic activity. And so one of the things that Divya has done is to take viable slices of tissue from the kidney or from the tumor, embed them in agarose, slice them thinly, and then culture them ex vivo for a period of days or even out to a week or two. They maintain metabolic activity and allows you to compare and contrast the kind of metabolic activity you observe in the intraoperative experiment versus in culture. And this is just to show that that exact same metric is retained in these viable tissues, which suggests that this is an intrinsic property of the tissue and not simply a consequence of the way the surgery was performed. So that's reassuring. Okay, Celeste mentioned acetate before. Acetate is a useful tracer because it allows C13 that starts on an acetate molecule that's being infused into the patient to enter into the TCA cycle without going through the PDH reaction. So if the entire problem of this change in labeling in the TCA cycle had to do with PDH suppression, then acetate labeling should be essentially acetate dependent labeling should be normal because it's not related to the PDH reaction. Okay, and so if you infuse acetate and now look at a downstream metabolite, in this case glutamate, indeed the acetate does not have any problem getting into the TCA cycle, at least not nearly as much as we observed with C13 glucose. Okay, but there's a subtlety of carbon-13 labeling that I think we have not probably paid enough attention to over the years. And that is that as the cycle progresses, the position of the carbon-13s in these molecules changes, okay? And in most conventional mass spec analyses, we just ignore that, okay? But actually, that's leaving a lot of data on the table. For example, in this acetate experiment I just described, when acetate enters the cycle, if you're looking at labeling in glutamate, you get 2 carbon-13s, but you get them specifically in positions 4 and 5 of the molecule. If that carbon-13 stays in the cycle, turns over one time, you still get N+2, but it ends up in a different position of the molecule. So the vast majority of mass spec data that you'll see in the literature is what's called an isotopolog analysis, where you're just counting up the number of carbon-13s in the molecule of interest. Glutamate has 5 carbons, so therefore there are 6 labeled forms, N+0 through N+5. But actually, the number of labeled forms of a molecule is not the number of carbons plus 1, it's 2 to the number of carbons. So actually there are 32 or 2 to the 5th labeled forms of glutamate, most of which we just ignore by grouping all of the N+2s together, but there's actually 10 different types of N+2. So you can get at most of this data with NMR spectroscopy. The problem is that the lack of sensitivity of NMR means you would need a fragment of 100 or 200 milligrams. And so what Feng Sai in my lab decided to do is to use modern mass spec techniques, capitalizing on its sensitivity, to take these informative molecules, break them apart in different ways, okay, and then use the fragmentation patterns in the mass spec to determine not how many carbon-13s are present in each molecule, but exactly where they are. And I'll just show you that his technique, when compared against the gold standard, which is NMR, actually not only works well, I think it works better. Okay, this is showing you the high level of correlation of labeling forms in 2 different molecules related to the TCA cycle, aspartate and glutamates, and how well they match up with the gold standard, which is NMR. The truth is, though, that with NMR, you only get a subset of these isotopomers, whereas in Feng's mass spec technique, he gets all of them, all 32 here, all 16 here, using only 1% of the tissue. So this is much, much more sensitive and more informative than what we've been able to do with NMR spectroscopy. Okay, so that allows us to ask some interesting questions about labeling in these TCA cycle intermediates. And so what I'm showing you here is Now, not just the acetate-dependent labeling in glutamate, but where the label is. I showed you before that if you just ask all of the M+2s, they're basically exactly the same between the kidney and the tumor. But if you ask the contribution of the first turn of the cycle, where the labeling is specifically in positions 4 and 5, now it's much higher in the tumor than it is in the kidney. Basically, what that means is that when we see labeling in the TCA cycle, in these clear cell renal cell carcinomas, it's mostly labeling the first turn. And the tumors do not seem to be very good at turning the cycle over into subsequent turns, okay? They lag behind the kidney in being able to do that. And based on what I said before about the usual connection between the electron transport chain and processivity of the TCA cycle, you might guess that that's because the mitochondria from the tumors are not respiring as well. And so to test that possibility, Divya did this experiment where now, rather than using carbon-13, in the surgical samples, she extracted metabolically active mitochondria, purified them, and measured their respiration in the lab. And she did this from patient-matched kidney and tumors from the same patient. And then in the respiration experiments, what you can see here is that all of these different components of the electron transport chain are suppressed in the mitochondria from the tumors relative to the patient match adjacent kidney. And so that's telling us, I think, that although it probably is absolutely true that there's some suppression of pyruvate dehydrogenase, just as all the preclinical studies said that there should, there's more to it than that. And the clear cells really seem to have a more fundamental problem with turnover of the TCA cycle. And that is related in part to their poor respiration. Okay, now, maybe we should have anticipated that based on this nice paper from Ari and Ed at Sloan Kettering several years ago. What Ed did in this paper is to figure out a way to use deep sequencing to quantify the amount of mitochondrial DNA in the tumor relative to the adjacent tissue. And he did that for multiple different types of cancer. You can see clear cells here down at the very bottom, okay, with on average, the least amount of mitochondrial DNA in the tumor relative to the adjacent kidney. Okay, 13 protein components of the electron transport chain come from the mitochondrial DNA, and therefore having less mitochondrial DNA should be correlated with less respiratory capacity and less oxidative phosphorylations. I think that's what we're seeing here. And it's reassuring to us that if you look at lung adenocarcinoma, which comprises most of our lung cohort, these are actually the highest. And so, and that may explain part of the reason why we see enhanced TCA cycle labeling in those tumors. All right. So,
Another
yeah,
quick
go ahead.
So one thing we've noted for years, and I was just curious if you see the same, it's not just that, they may have fewer mitochondria or less mitochondrial DNA. They have aberrant looking mitochondria. They just do not look normal in terms of all their structures and et cetera. Do you guys see the same thing in your cohorts?
We haven't looked at mitochondrial ultrastructure very carefully. And so my guess,
you can see really strange structures, at least to me. And I think this is something that Amato Giacchi appreciated quite some time ago too.
Yeah, I think we need to look at that more carefully. I mean, because at this point, Celeste, we know many of the molecular mechanisms that account for things like Christie formation.
Mm-hmm.
And, as you'll see, and I think you've seen us present some of this data before, there's something very different about the activity of these treatment naive localized tumors and the ones that go on and metastasize that are suggesting a change in mitochondrial activity that we have not been able to pinpoint through gene expression or mitochondrial DNA content.
Mm-hmm.
So something about maybe the structure of the mitochondria that would potentially be recoverable in a way that could be independent of the mitochondrial DNA content would be really appealing. So I'd love to talk some more about that with you.
Okay, we have a beer coming up, so we'll work that into the agenda.
Okay, good. That's great. I'll just comment one more thing. I'm going to wrap up here in a minute or so, but that Ed and Ari made the very interesting comment in this paper that despite the depletion of mitochondrial DNA in the kidney cancers, the patients that did the worst in their study were the ones that were sort of towards the right on this distribution of mitochondrial DNA. And so with that in mind, we've tried to do the carbon-13 infusions in patients that had either synchronous or asynchronous ccRCC metastases. And I'm showing you data here from about the 10 or so patients that we've studied. It's quite striking to us that the labeling of the Krebs cycle intermediates appears to recover. Okay, here's the nice suppression that you see between the adjacent kidney and the primary tumor. Okay, and here are 10 patients with ends for each metastatic site shown here that were infused usually later. Okay, so these patients with the 3 lung mets, we thought these patients had primary lung tumors and then went in and found out they actually had metastatic ccRCC. These 2 adrenal mets here were synchronous metastases. So the patients were infused during the same operation when the primary tumor came out. So we were able to compare it directly. But in every case, we see Krebs cycle intermediate labeling that really seems to be as high or higher than the adjacent kidney and much higher than the primary. Okay, so I do not have a mechanistic explanation for why this occurs yet. It does not seem to be related to the content of mitochondrial DNA or related to the abundance of mitochondrial transcripts. It suggests there's something else happening in these metastatic tumors that is appealing because it suggests to us that there is something important about having oxidative phosphorylation and TCA cycle activity that maybe is being selected for in these mets. And so just one last piece of data is to say that we are trying now to do a gain of function experiment to ask if we drive mitochondrial activity in the VHL deficient state, does that do anything to metastatic spread? So what we've done is to go into a very simple model, 7860 cells, VHL deficient, long established clear cell line, and put this NDI1 protein into those cells. That's the one that allows you to recycle NADH to NAD plus and have some electron transport chain activity. You can see that drives respiration. And it basically resets this citrate pyruvate labeling ratio in culture. And we're just getting into in vivo experiments now. But what this simple experiment shows is that if you inject control cells versus NDI1 expressing 7860 cells into the tail vein of mice, you get significantly higher metastatic burden when the only thing that's different is the expression of NDI1. And so, we have a lot more work to explain why this is happening, but it does suggest that, having a little bit more activity of the electron transport chain may be important even for VHL deficient cells. So I'm going to have to skip the last part about glutamine metabolism, but we can talk about that another time if you'd like, and the different ways that could potentially be targeted. And let me just sum up by saying that I think the carbon-13 infusions can be useful, certainly in mice, but also I think in patients. And most relevant to clear cell is to say that as expected, they display reduced TCA cycle labeling and oxidative phosphorylation seems to be fundamentally caused by a number of metabolic abnormalities in the mitochondria. But the metastatic tumors seem to regain the ability to oxidize glucose, at least as we read it out through these intraoperative carbon-13 infusions. We have lots of questions that need to be addressed now. I think in terms of the metastatic phenotype, the most pressing question to me, both for the lung cancer study and the kidney cancer study, is what is it about oxidative phosphorylation that's important? And would there be any kind of feasible way to silence those benefits without having to do something as potentially toxic as just shutting down the electron transport chain, which systemically I think is probably not going to be tolerated very well. All right, let me thank the people who did the work, most importantly for the human infusions, Brandon and Divya. We have great collaborative teams here, both in lung cancer and kidney cancer. I didn't show any data on the orthotopic tumor grafts developed by Jim Brugarolas, but it's a great value to us to have access to those. And I can take questions for a couple of minutes. I'm sorry if I went too long.
Yeah, Ralph, this was great. Thanks. Yeah, I know you got a tight timeline, but maybe have a time for a minute or two of questions.
Please.
Sure. I know we asked some throughout. Anybody else have questions?
I'll follow up with you, Ralph,
ahead.
offline, but a very quick thought. The reductive carboxylation that typically we see with that Othon and George have mentioned about glutamine being taken up into citrate and the lipid pathway in CCRCC. I was thinking it's kind of interesting that your lung cancer lines with EGFR activating mutations also seem to phenocopy that same phenomenon. And so in my mind, one idea that came up is there's literature around how EGFR is activated in VHL deficient tumors, in part because it escapes proteolysis. People have in fact suggested that EGFR might be a direct ubiquitinated target of VHL, but also transcriptionally probably being induced. So there's no mechanistic understanding about why or how reductive carboxylation is occurring in CCRCC. But a quick experiment would probably be to try the EGFR inhibitors and see if we now can dampen the flux through that in that direction.
But that is a great thought. And so I've presented this before, so you may have seen this before, but in the VHL deficient tumor grafts from Jim, they all have this signature of reductive carboxylation in vivo in the kidney. And that is dependent on glutaminase because we can shut it down with CB839. I have assumed that is because of the suppression of pyruvate dehydrogenase in a VHL loss dependent fashion because of the work that Othon did years ago, showing that actually, seems to be sufficient to drive that pathway. But what we've all lacked are post-translational modifications on IDH1 and IDH2 that would explain that. And so, there's no reason, these are not mutually exclusive. You can have a suppression on PDH that reduces the amount of acetyl-CoA that comes from the PDH reaction and concurrently have a change in EGFR signaling that is allowing you to adapt to that situation and make a pool of citrate from reductive carboxylation. So we'll try the experiment. Thanks for the advice.